All right, thanks for the invitation and thanks for the introduction. Most people say that they get busier after retirement, and I did that, so I took on several other jobs. So, but it's fun to come back and address some research that I did in collaboration with programs at Western Illinois University and Southern Illinois University, based on a SARE grant that we got a few years ago. And with the interest in cover crops that we've been seeing, there's using cover crops for various things. I'm a plant pathologist, was a faculty member at the University of Illinois for almost 30 years, and my specialty is in soil-borne diseases and sort of the impact of environment on disease development. So I thought, we're seeing the use of cover crops for weed management and for crop growth and soil health issues. What kind of an impact are these going to have on disease development, either increasing disease or possibly suppressing diseases? A fair amount of work's actually been done with vegetable crops and the input of organic matter or the use of cover crops to try to suppress or manage diseases in vegetable crops. But surprisingly little has been done with field crops, corn and soybeans in particular. Some of that is just because adding organic matter to a smaller acreage system is more economically feasible. But cover crops, we're now using them on field crops as well. So this project aimed at seeing how that impacted the disease development. Now this was in a corn soybean system where we planted the fall cover crops after the corn came out, and then followed that with the soybean crop. So we're looking at it on soybean, the impact on soybeans in a corn soybean system. So some of the benefits of using cover crops are reducing soil erosion, reducing soil compaction, nitrogen scavenging if you're using the grasses, or adding nitrogen if you're using the legumes, increasing soil organic matter, which helps with a number of factors, increasing water penetration and retention of nutrients and so forth, and looking at weed management. And so again, the goal of this project was, what is it doing to diseases, in particular soil-borne root-infecting diseases? There's a phenomenon in plant pathology called the disease suppressive soil effect. And this is where the environment, or there could be microbial changes in a soil that make it less likely that a disease will develop. Even if the pathogen is there at the same level as it would be in a conducive soil, it could be physical factors, or it could be biological factors that just don't allow that pathogen to cause the same amount of disease as it would otherwise in a conducive soil. All soils have some natural disease suppressive capability. So if we take a soil, for example, out of a field and measure the ability of a pathogen to cause disease, and then we take that same soil and we sterilize it, we put it in an autoclave or what have you, and then put the pathogen back in, we'll get more disease because there are fewer microorganisms in there, fungi and bacteria and other things that would normally keep that pathogen from being active, or sometimes, in some cases, it's stimulating resistance in the plant. So the bacteria and fungi that are in the soil stimulate host resistance. So if you remove those factors, that soil becomes more conducive. But there are things that we can do to soils that make them more suppressive. So again, this has been done a lot with vegetable crops, and so we wanted to see if we could do what impact cover crops have. Now, in that first slide, I said that this was actually a cooperative venture between three institutions, University of Illinois, Western Illinois University, and Southern Illinois University, and we did the studies at in all three of those regions. But for various reasons, the data from Southern Illinois didn't make it into the analysis for the talk that I'm giving you today. So it's focused on two regions, East Central Illinois, to where the University of Illinois is, and Western Illinois University is in Macomb. In both of those locations, we had both on-station, so the university plots, and then we also had some on-farm locations. So the studies that we were doing, we did in 4 different locations over 3 years. We looked at 4 cover crop species, cereal rye, rapeseed, canola, and then a mustard that was selected because it specifically was advertised as having anti-soil-borne disease properties, so high levels of a compound called glucosinolates that would make it, once it got into the soil, it would suppress diseases. So the collaborators, again at Western Illinois, Joel Groover and Steve Ayers was our farmer in East Central Illinois, and Brad Hunt was the farmer in Western Illinois, and then former graduate student of mine who is now at the University of Wisconsin was also involved with this study. So these are the cover crops that, that we were using. The plots that we had on the university campuses were fairly small, 10-foot wide, so basically 4 rows by 40-foot long plots. The farmers wanted something more field scale so that they could plant the cover crops and deal with them with the equipment that they had. So they were larger plots, 60-foot wide by 1,000 feet long. And on the on-farm plots, we limited it to just 2 of the cover crops, the cereal rye and the rape, and then had the fallow as the control. So looked at all 5 treatments in the university plots, and then 3 treatments in the on-farm plots. So the cereal rye was generally performed very well in all the locations. Again, we're planting in the fall as soon as we could get the corn crop off and get the cover crops planted. That was, for the rye crop, that was plenty of time. We were typically planting somewhere between the middle of September and sometime in the first week or so of October. And that was enough time for the rye to get established and do fairly well. In some cases, the rye got very well established and didn't get controlled as early as we wanted it to. So in one case in particular, the rye got pretty big here. You can see that rye is over 3 feet tall. And we were planting soybeans into it, but surprisingly it did very well. So our protocol was to, once the rye got between 12 and 18 inches, it would be terminated and then incorporated or tilled into. So the 2 on-farm trials we had, both of our farmers were no-till farmers, and so we didn't incorporate the cover crops in those situations, whereas the university plots, the cover crops were actually incorporated into the soil. In this particular case, this was one of the on-farm trials where the farmer wasn't able to get in because of rainfall issues, and the crop got pretty large before he could get in and terminated it. But amazingly, even when he no-tilled into that pretty substantial stand of cereal rye, the soybeans came up just fine, and by August you couldn't tell the difference between the rye cover crop plots and the fallows. So I was really amazed that the soybeans did as well as they did coming through that stand of rye. Some of the other crops that we looked at, we did not get as consistent a stand with. The rye and canola, sometimes they established well, but other times they did not. It really depended on the amount of rainfall that we got after planting. And so if we got a good rain shortly after planting, again, planting end of September, beginning of October, they could get established and we would have a good stand the next spring. If we didn't get much of a rainfall, then we didn't really get a very good stand establishment. This is what we might see in the spring if we got good stand establishment. The mustard was not winter hardy in Illinois, and so typically we would get a small stand of mustard and then it would quickly kill after the first frost or two. And so I wasn't really, after seeing how it performed, I really wasn't expecting to see very much in terms of responses to that, but we kept it in the study anyway. So the things that we were evaluating, we looked at cover crop biomass in the spring. So we didn't take any measurements in the fall, but we waited a few weeks before planting to take cover crop biomass in the spring. We would then terminate it, and again, in the university plots, we would incorporate it into the soil. And then we'd plant soybeans in there 2 to 3 weeks after termination of the cover crop. We'd take soybean stands, so looking at the emergence of soybeans, and then look at disease levels that developed both early in the season, looking at seedling diseases in the V3 to V4 stage, and then later on in the reproductive stage, we would look at both foliar and root diseases to see what kind of diseases were developing, and then we would take yields at the end of the season. We also took soils and took those into the greenhouse and did what's called a bioassay. For some reason, the farmers didn't want me to inoculate their fields with pathogens so that we could study the level of disease that would develop. So we had to do that in a different way, and we did that by collecting soil in the field right about the time of planting, and then we would take that into the greenhouse, inoculate it with 2 different soil-borne pathogens, and then plant a susceptible soybean into it, and to see if that soil had developed any kind of disease suppressiveness as a result of the cover crop treatments. We also then looked at the egg counts of the soybean cyst nematode to see if we were having an impact on that. And then we also extracted the DNA from those soils to look at specific populations of soybean pathogens, and then to do just a general analysis of the microbial community structure of that soil, because we wanted to see if we did see a suppression, could we attribute it to some change in the total composition of bacteria and fungi that would be responsible for that suppression. So some of the common diseases of soybeans that we did that DNA analysis for included sudden death syndrome caused by Fusarium virgiliforme, Rhizoctonia root rot caused by Rhizoctonia solani, anthracnose caused by Calototricum, SCN or soybean cyst caused by the soybean cyst nematode, brown stem rot caused by a Phyllophora, Phytophthora root rot, charcoal rot, and Sclerotinia stem rot. So we did DNA analysis of the DNA that we extracted from the soil for most of these diseases. The diseases that we evaluated in the field, again, we couldn't inoculate, so we just had to depend on the diseases that developed there. So foliar diseases, we consistently saw Septoria brown spot and a bacterial blight. And then in roots, we consistently saw some sudden death syndrome and Rhizoctonia root rot. The one exception in terms of inoculation was on the University of Illinois plots. I could inoculate those plots. So we actually expanded the study in the university plots at the University of Illinois and had non-inoculated plots that we inoculated with Rhizoctonia solani, the Rhizoctonia root rot pathogen, and plots that we inoculated with Fusarium virgiliforme, the sudden death syndrome pathogen. So we could increase the level of disease that we evaluated in those plots. For the greenhouse bioassay, we looked at to collected soils from the different cover crop plots, and then inoculated those soils with either Rhizoctonia or Fusarium, and then planted with a susceptible soybean variety and measured the amount of disease that we saw with them. So the bioassays we did by growing up the pathogen in culture, and then we actually blended up colonies of it and mixed it with sand, and then add a known volume of fungus per sand, and then we use that sand to mix it in with the soil. So we had a quantified amount of pathogen that we added to the soil, put it into small containers, and then put a susceptible variety of soybean, let it grow for a certain amount of time, pulled those plants out, and then rated them for disease development. So this was with Rhizoctonia. We did something similar, had slightly different methodology, but for the Fusarium, for looking at sudden death syndrome. So we looked at those two diseases in the greenhouse bioassays. For the DNA analysis, these are the disease pathogens that we looked at. We had to do them in a couple of different runs in order to get them all in there. But fortunately, we have a colleague of mine who had developed all the assays for doing that. And then using, again, taking that total soil DNA, we looked at the general microbial community structure. In terms of biomass, it varied from year to year depending on how soon the crop got planted and what the environment was like after planting. Generally, the cereal rye did very well, as you can see in most of the plots. I don't know if that's cereal rye was generally generated the most biomass, differed from year to year depending on how much rainfall we got after planting. The mustard generally, again, we're taking these measurements in the spring. So the mustard, there was only one spring in which we still had a mustard crop left. In most of the locations in most of the years, it winter killed and there was nothing left in the spring to evaluate. So if we, yeah.
So cereal rye no-till only grew in one plot?
So that was at the Western Illinois University on-campus farm, and that's a organic certified farm, and the person who was doing it wanted to expand our specific treatments, and he wanted to do a no-till with rye. It wasn't in any of the other ones. It was just because that particular cooperator wanted to do that. And so we said, sure, put it in. So looking at soybean stands following any of those treatments in general, we did not see any significant difference between the cover crop treatments and stand, except for the plots at the University of Illinois where we inoculated them with Rhizoctonia solani. And then we did see a difference where we saw a significant increase of seed germination in the rye plots that were inoculated with Rhizoctonia as compared to the fallow plots where we inoculated with Rhizoctonia. So when we didn't inoculate with the pathogens, everything looked the same. But in the plots where we actually added the fungus in and had a fairly high amount of disease, then we did start seeing some differences in emergence or in stand establishment where we got some improvement of stand establishment as a result of, in particular, the cereal rye crop. And I'll show you some results there. So this is the stand establishment in 2011 from those three subplots within the treatments that we put on in the University of Illinois location. Under the control treatment, that was no pathogens added. You can see there were slight variations, but nothing that was statistically significantly different. They were all had the similar levels of emergence. Same in the plots that were inoculated with the Fusarium, trying to establish sudden death syndrome. We didn't see any differences in emergence and stand establishment there. But in the plots that we inoculated with the Rhizoctonia solani, we saw an amazing amount of disease. And this wasn't only in my plots, it was just the environment in that particular year was particularly good for Rhizoctonia. And a colleague of mine who was also looking at Rhizoctonia plots, he was looking at fungicide treatments. His plots were devastated also where he inoculated. And so you can see in the plots that were fallow, or the mustard plots, again, the mustard, we didn't have much cover crop development in those plots, very low stand establishment there. And in fact, it was hard to find plants in those plots. Whereas in the rye plots, the amount of seedlings that emerged was almost the level that we saw in the non-inoculated plots. So it was just amazing. And I actually have photos of that here. So you can see on the left-hand side, those are the fallow plots that were inoculated with Rhizoctonia, and just a seedling or two here and there showing up. The rape was sort of intermediate, so we got definitely a significant improvement over the fallow plots, but in the rye plots there was essentially no loss of plants as a result of Rhizoctonia. And I thought, this was the first year of the study, and I thought, this is amazing. It's going to be easy to show differences. And then, of course, the next 3 years, I didn't see anything like that. You know, the level of disease that we saw in the following years just wasn't as high as it was there. Yeah?
Let me ask, on the university plots, so these end up being a continuous soybean rotation where you were doing the study, or?
So they put us in a specific location. For anybody that's doing plant pathology, inoculating soil work, they say, those are your plots over there, and that's where you get to be. So I mean, they're not only ours, so they rotate, you know, the weeds people are in there and other people, but whenever we're going to do inoculation, we have to go to a certain area to do that so that we're not inoculating everybody else's fields. So, yeah. No. So in this area, we have established a corn-soybean rotation. Yeah. So it's always corn following soybeans. But yeah, there are definitely plots where we've been looking at sudden death syndrome, for example, and we can go back there and regularly get the disease because we've inoculated it so often. So. So this is just to say that this slide shows that we didn't see that level of improvement every year. So in 2011, when we looked at stand counts, in most places there were no significant differences because we weren't inoculating. The disease levels weren't very high. But in the UIUC, that's the University of Illinois plots, where we added the Rhizoctonia, inoculated the plots, and we saw highly significant effects of the cover crops. We didn't see any effects in 2012, but in 2013, again, where we inoculated with Rhizoctonia, we saw significant effects of stand establishment with the best establishment in the rye cover crop plots. We saw an effect also in the western plots, again, also in 2013. Only that way it was sort of the other way around. In the mustard plots, we saw some reduction, and we didn't see a reduction in the rye plots in that case. So some variation from year to year and location to location. So here's what we're rating for at the seedling level. So this is a few weeks after emergence. We would dig up the plants and rate the seedlings for Rhizoctonia root rot. These brownish lesions on the plants are where the Rhizoctonia is infecting, and we would just measure those lesions. So from 2011, in the plots that we inoculated, we didn't have enough plants left to dig out, or we wouldn't have had anything to evaluate later on in the season. So we didn't do the Rhizoctonia root rating in the inoculated plots in 2011, but we did it in 2012 when we had a better stand establishment. And as you can see here, we're seeing some similar results in that we're getting the greatest growth of the Rhizoctonia in the fallow plots and the smallest lesion size, a suppression of Rhizoctonia in the rye plots, and then the rape and canola is somewhere in between. We also looked at effects on Rhizoctonia in those greenhouse bioassays. Again, I couldn't go and inoculate all the other sites, so we didn't see a whole lot of Rhizoctonia developing in the other locations, but we could do it in the greenhouse. And again, it would vary from year to year and from location to location. So again, you can see that in 2011, the bioassays didn't really show much significant difference, but typically we would see sort of a trend of some higher levels in the fallow and lower levels in the rye, but not always. In 2012, the bioassays did show an effect of cover crops, again with the highest levels of Rhizoctonia in the fallow plots, the lowest levels either in the rye plots or in the rape plots. So it's backing up what we saw in the field studies. And then in 2013, again, we saw a pretty good effect of the fallow, showing the most level of disease, and the rye and the rape. And for some reason in this particular year, the canola showed pretty good disease suppression as well, but we didn't see that in most locations in most years. We also looked at evaluation of sudden death syndrome. We never saw, even in the plots that we inoculated in the field, we never saw a difference of cover crop and the level of disease that developed for sudden death syndrome in the field. But we did see some in the bioassays. Not in 2011, but we did see in 2012, again with the rape, and in some cases the rape and the rye, both causing a suppression of sudden death syndrome. So again, not constant, not every year in every location, but we never did see more disease in the cover crop plots than the fallow. So the fallow, if we ever saw a difference, it was always that the fallow was the highest level of disease, and then the cover crop plots were lower. Again, this is in the greenhouse. We did see some disease suppression of the fallow versus the rye. For soybean cyst nematode, however, it was fairly consistent that we would typically see higher cyst counts in the fallow plots and lower cyst counts. So this is extracting the eggs of the soybean cyst nematode from the soil and counting them. We saw higher counts in the fallow plots and lower counts in most of the cover crop plots. Mustard, again, mustard never really established very well, so we typically didn't see a lot with that. But with the rape and the rye and sometimes the canola, we did see differences. So that's 2011, 2012. Again, fairly consistent with effects of soybean cyst nematode, seeing a suppression with cover cropping. Now this is the results of that DNA community analysis, where we're hoping to see, to say, you know, when we planted with rye or when we planted with rape, it changed the community structure, and then we could use that to say, okay, let's go find what is actually doing that suppression. So this analysis here was an analysis looking at the community structures at the different locations. So each of those, the different colored triangles there, those samples were taken from from those locations. So the green triangles are from the farmer plots in eastern Illinois. The purple triangles are from the farm plots in western Illinois, and so forth. And then we did this analysis called an ERISA analysis. Again, looks at overall community structure and gives us some measure of that. And we could say, okay, yeah, that sample was from that location because it had this community profile, and this sample was from this location. But when then we tried to take it and look at the community profiles based on the cover crops that were planted there, we didn't see anything. No pattern emerged. So, so although the test worked well in it being able to characterize community profiles from different locations, it didn't show us that there was a difference associated with cover crop, particular cover crop treatments, which is what we were hoping for. Now looking back on it, and after talking with other researchers who do this, I guess it shouldn't be a surprise, because we were taking these samples shortly after terminating the cover crop, and it was only one season of cover cropping, and most of the farmers who work with it tell you, you have to cover crop for 3 or 4 or 5 years before you see a consistent effect. And we probably just didn't do it long enough, and it was only a few weeks after termination. Yeah?
You were in a corn-soybean rotation. Did you do a cover crop after the corn?
So this is just looking at effects on soybean after one fall cover cropping, and it was a new location every year. So it wasn't a repeated study, and I think if you're going to see a difference in microbial profiles, it's not something that's going to happen instantaneously. It's probably going to take several years of cover cropping, because microorganisms in the soil are amazingly good at suppressing changes. They buffer themselves, and that's one of the things that helps prevent pathogens from coming in and taking over. It's because of the organisms. So it's not easy to change them, and it's going to happen over a long time. So if we could do plots after 5 or 6 years, we might start seeing some differences. Looking at yields, the only place where we really saw a yield difference was where we inoculated with Rhizoctonia, and obviously there we had such dramatic reductions in stand that we saw dramatic reductions in yield as well. So in the plots, oops, back up. So on the, where am I going here? Not sure what happened there. So in the plots here where we inoculated the field plots with Rhizoctonia solani, the fallow, again, very poor stand establishment resulted in fairly poor yields. Again, the mustards, we had very poor stand establishment there, whereas in the rye treated plots, very good stand establishment and resulting in substantial increases of yield over the fallow plots. In all the other locations, essentially no effect of cover cropping on yield, which is a good thing from the farmer's perspective. It's saying they're not getting a yield reduction in soybean as a result of cover crops. If those cover crops then are providing benefits like controlling soil erosion, adding organic matter, they're not going to take a yield hit as a result of it in the soybean crop at least. Now I know that there's a difference with corn. On corn, there do seem to be some yield hits with some cover crop treatments. But soybeans, it seems to be okay. So my take-home messages from this study are that both rye and rape appear to have some ability to cause some disease suppression. We did see the highest stand establishments with those 2 cover crops, with rye being the best, that works best at least in the situations that we were using. They have the potential to induce some disease suppressiveness in the soil that we could measure in bioassays in those greenhouse studies for both Rhizoctonia and in some cases for sudden death syndrome, although it wasn't as continuous or significant as what we saw with Rhizoctonia. All of the cover crop treatments that we looked at, we saw some suppression of the soybean cyst nematode. As far as we could tell, based on our DNA analysis, it really didn't affect any of the other diseases that we were monitoring. And cover crops didn't really result in any difference in microbial community structure, at least in the study that we did.